Hello viewers, welcome to the course Elements of Genetics. We shall try to understand linkage and crossing over, their types, mechanism and significance. Beginning with some exceptions to Mendel's laws of inheritance. Let us appreciate that hitherto we had been trying to understand that Mendel proposed certain laws of genetics and the future researches confirmed the Mendel's laws despite the fact that Mendel was not aware of the existence of the chromosomes. In chromosome theory and in cell division, we observed that the later researches in cytogenetics proved that even the behavior of chromosomes were in support of Mendel's laws of inheritance. In today's part, we begin with certain exceptions that researches which did not conform with the Mendel's ratios, but let us agree to believe that these exceptions did not prove the Mendel was wrong, but these exceptions rather proved the rule. Based on the scientific contribution of Morgan and others that led to the discovery of genetic linkage, the chromosomal exchange of genetic material via crossing over of chromosome during meiosis and how this promotes genetic variability, and to construct genetic maps from crossover data. The discovery of genetic linkage is basically the presence of genes on non-homologous chromosomes which assort independently, and the genes on the same chromosomes may instead be inherited together and belong to a linkage group. A very important point emerges here that Mendel could establish certain laws of dominance, laws of independent assortment and law of segregation because the traits he used were either single or whether the traits were in pair, they were located on different chromosomes. But what happens when the characters or the traits are located on the same chromosome? These characters may be linked and therefore they do not assort independently, and therefore ultimately we do not find exactly the same ratios which were proposed by Mendel. This is the situation which is known as the linkage, and because of this fact that large number of genes are present on the same chromosome, the chromosome is also known as a linkage group. The classical genetics analyzes the frequency of allele recombination in progeny of genetic crosses. New associations of parental alleles are recombinants produced by genetic recombination. Test crosses determine which genes are linked and a linkage map, that is genetic map, is constructed for each chromosome. Genetic maps are useful in recombinant DNA research and experiment dealing with genes and their flanking sequences. The linkage and recombination word come together. Let us try to recall with the facts of meiosis, particularly during prophase 1, where the chromatids they twined and entwined over each other, and there were chiasma or chiasmata formations from the point where the genetic segments were exchanged. This is known as the recombination. The current high resolution maps include both the gene markers from the test crosses and DNA markers composed of genomic regions that differ detectably between individuals. The gene marker concept and the DNA marker concept is an advancement in genetics which is based on the linkage and crossing over researches. Linkage in the sweet pea. There are certain now examples of the linkages that when genes are present on the same chromosomes, they are linked to each other and they do not separate or assort independently as per the predictions of the Mendel's law. Bateson, Saunders and Punnett in 1905 found exceptions to the law of independent assortment in their studies of pea plant. They crossed two true breeding strains, one that produced purple flowers and long pollen and another that produced red flowers and round pollen. In the expected result of F1 into F1 cross, if genes for flower color and pollen size were to assort independently. But in F1 generation, the diploid and genotype, that is capital P capital L small l phenotype, and the purple flower and long pollen. The expected genotypes of possible F1 gametes haploid should be all 1/4 capital P L, capital P small p capital L, and small p small l. That is the result of the expected test cross, which should have been as per the predictions of the Mendel, that all should fall in ratio of 1:1:1:1, that is 25% if the total number of offspring is 100. The expected phenotypic ratio of F2 generation, that is purple long, purple round, red long and red round as shown here, is 9 out of 16, 3 out of 16, 3 out of 16 and 1 out of 16. It is very famous law of independent assortment or dihybrid cross, where the ratio we always observe in 9:3:3:1. But in the experiment, the scientists observed a different ratio, that is 69.5, 5.6, 5.6 and 19.3. These observed results indicated partial linkage of genes. Dominant purple and long characters occurred together more often than predicted, and therefore the F1 haploid gamete in test cross came out to be 44%, 6%, 6% and 44%. So these deviations from the expected result of the Mendelian genetics are in fact indicators and suggesters of the existence of the linkage, meaning otherwise that the traits of inheritance are located on the same chromosome, they are linked together and they move together and they do not assort independently as per Mendelian predictions. Now Morgan's linkage experiment with Drosophila. Let us agree and appreciate that most of the linkage research has been contributed by the fruit fly research by Morgan and Muller. Both the white eye gene W and a gene for miniature wing M are on the X chromosome. X chromosome is the sex chromosome. Morgan 1911 crossed a female white miniature with a wild type male. In the F1, all males are white eyed with miniature wings and all females are wild type for both eye color and wing size. Morgan concluded that during meiosis, alleles of some genes did not assort independently but assorted together because they lie near one another on the same chromosome. Here we appreciate that the importance of chromosome theory and the importance of meiosis is reconfirmed in the sense of the linkage and the crossing over. F1 interbreeding is the equivalent of a test cross for these X-linked genes, since the male is hemizygous recessive. In the F2, the most frequent phenotype for both sexes are the phenotypes of the parents in the original cross: white eyes with miniature wing and a red eye with normal wing. Non-parental phenotypes occurred in about 37% of the F2 flies, below the predicted 50% for independent assortment. This indicates non-parental flies result from recombination of linked genes. In these experiments, the ingenuity of scientific deductions is very apparent in the sense that whatsoever deviated from the Mendel's law were interpreted to establish certain other laws of genetics which are contributing continuously in the advancement of the genetical sciences. Morgan experimental crosses of white eye and miniature wing variants of Drosophila melanogaster is shown in this particular diagram, which has been described previously. Morgan's linkage experiment with Drosophila suggests, and Morgan proposed, that during meiosis, alleles of same genes assort together because they are near each other on the same chromosome. Recombination occurs when genes are exchanged between the X chromosome of the F1 females. Now, this is the ingenuity of the drawing scientific deductions and the conclusions whenever there is any deviation in the predicted ratios occur. And the terminology which used now in case of crossing over, linkage and recombination, that a chiasma, we also came through this word during meiosis or prophase 1, the plural is chiasmata, is the site on the homologous chromosomes where crossover occurs. Crossing over is the reciprocal exchange of homologous chromatid segments involving the breaking and rejoining of DNA. Crossing over is also the event leading to genetic recombination between linked genes in both prokaryotes and eukaryotes. The chiasma is a point where the chromosome or chromatid meet, and at that point of juncture of meeting they break and fuse, and therefore with this mechanism they exchange the genetic material by which the progeny is different from the parent in their genetic constitution. Now, this is the mechanism of crossing over between homologous chromosomes, and we see that the diverse chromosome from each parent, that is male parent and female parent, they come together. A chiasma point is formed, say here in a yellow chromosome and the blue colored chromosome. Then there is a crossover point in the middle diagram we can see where breakage and crossing over is taking place, and in the third figure there is a reunion and exchange is complete, and therefore we see a new recombinant type of chromosomes are produced after chiasma formation, after crossing over, after breakage and fusion, and that is why the meiosis has a great role in linkage and crossing over. The gene recombination and the role of chromosomal exchange has also been explained in a corn experiment that Creighton and McClintock in 1931 performed crosses with corn plants and found cytological evidence that crossing over occurs during meiosis and is associated with the physical exchange of parts between homologous chromosomes. It is again and again reminded that the exceptions to the Mendel's law, which came out as the theories and ideas and concepts regarding linkage and crossing over, is a result of understanding and knowing the complete structure and function of chromosome as well as the behavior of chromosome during mitosis and meiosis, particularly in meiosis, because this division takes place in the reproductive cells or the germ cell, and therefore is responsible for creation of new genetic constituents in the offspring. The study used a corn strain heterozygous for two genes on chromosome 9. One gene determines seed color, capital C for colored seed, small c for colorless. The other gene is involved in starch synthesis. The wild type allele, capital W small x, produces amylose, and amylose plus amylopectin, that is normal starch. The Vexi mutant, small w small x, lacks amylose. In this corn strain, the appearance of each chromosome 9 homolog correlated with its genotype. It is important to note now that the scientists have now embarked upon not only the morphological traits but the biochemical traits which are responsible for certain expression at morphological level. For example, the color viscosity, the color behavior or the grain behavior. Evidence of the association of gene recombination with chromosomal exchange in corn is designated here in this diagram very clearly, where in A there is a heterozygote, there is a B the crossover occurs, and in C the recombinant chromosomes are formed after the crossing over. The same thing happens that in the chromosome 9, which has been shown here in the A heterozygote, there is a crossover, and after the crossover and breakage and fusion is complete, we can see the creation of a new recombinant or a new genetic constitution in the chromosome 9. The Drosophila experiment, they shortly after Creighton and McClintock published their result. Stern reported identical result for experiment done with Drosophila. He reported on two linked gene loci. From experiments such as these, it became apparent that genetic recombination results from physical crossing over between chromosomes. These all ideas suggest one common thing, that the phenomena of linkage and crossing over is common in the entire living kingdom. It is not an exclusive characteristic of a plant or an animal or a Drosophila or pea, but it is rampant everywhere. And all the experiment which has been done with the plant systems like corn or the pea or the animal system like Drosophila, they prove and provide evidence for the existence of linkage, recombination and crossing over. Crossing over at the tetrad stage of meiosis suggests that crossing over occurs at the 4 chromatid, that is tetrad stage, in prophase 1 during meiosis. In Neurospora crassa, orange bread mold forms 8 haploid spores. Their arrangement in the ascus reflects the orientation of chromatids in the metaphase tetrad of meiosis. To determine when crossing over occurs, crosses were made between haploid Neurospora strains of different mating types, capital A and small a. Now here we have seen that varied kind of experimental materials have contributed to the advancement of the understanding of linkage and crossing over. Hitherto we had been studying with the pea plant or with the corn plant or with the Drosophila, but now here is a fungi known as Neurospora crassa, which has a beautiful system of showing the meiotic division in the form of ascospores, and this cycle completely and perfectly shows that how Neurospora crassa, the two different kinds which has been shown with the yellow color as well as the blue color, how they mate asexually and sexually to show the phenomena of linkage and crossing over. Experiments showing that crossing over occurs at the 4 chromatid stage of meiosis in Neurospora crassa, as in the bottom we can see that 8 ascospores, they are the result of the new genetic recombinant after crossing over. Now, locating genes on chromosomes, the mapping techniques. Now, this is yet another advancement in the science of genetics that we have seen from the Mendel, the laws of Mendel. Then we came about to understand the chromosome theory. Then we knew about cell division, and the cell division led to the understanding of linkage and crossing over, and now that understanding of linkage and crossing over is giving a way to understand the genetical mapping or mapping techniques, and these involve detecting linkage through test crosses, gene mapping by using 2-point test crosses, generating a genetic map, double crossover and 3-point crosses. For developing genetic map, it is important to detect linkage through test crosses. To construct a genetic map or linkage map, the genes in question must be linked or located on the same chromosome. To test for linkage, a test cross is used. Test crosses are between one individual of unknown genotype and a homozygous recessive individual. The test cross is to show that two genes are linked. Concept of a genetic map. In an individual heterozygous at two loci, there are two arrangement of the allele, that is cis coupling arrangement has both wild type alleles on one homologous chromosome and both mutant on the other, W plus M plus and W and M. Trans, that is repulsion arrangement, has one mutant and one wild type on each homology, that is W plus M and W M plus. A crossover between homologs in this cis arrangement result in homologous pair with the trans arrangement and vice versa. Frequency of recombinant is the same, regardless how the alleles of the two genes involved are arranged relative to each other on the homologous chromosomes. Geneticists use recombination frequencies to make a genetic map. A 1% crossover rate is a genetic distance of one map unit is equal to 1 centimorgan. Genetic distances are additive. Mapping using 2-point test crosses. The percentage of recombinant resulting from crossing over is used as a measurement of genetic distance between two linked genes. In all cases, a 2-point test cross should yield a pair of parental types that occur with equal frequency and a pair of recombinant types that also occur equally. The value for percentage of recombinant, that is recombinant upon total of progeny into 100, is usually directly converted into map units. Now all these descriptions indicate only toward one thing, that the genetic maps are created using mathematical and statistical tools by calculating the crossover frequency or recombination frequency. The genetic map is generated, that a genetic map can be constructed by estimating the number of times a crossover event occurred in a particular segment of the chromosome. The map distance between two genes is based on the frequency of recombination between the two genes, and the recombination frequency between genes in chromosome can be computed as the percentage of progeny showing the reciprocal combinant phenotypes. And this diagram again shows the relationship between crossing over and the map distance. The distance is shown in the diagram between A, B and C genes, and therefore the infrequent crossing over in this region, that is why A, B genes are nearer to each other, and frequent crossing over in this region, that B and C genes are quite distant. For any test cross, the percentage of recombinant cannot exceed 50%. The unlinked genes, that is 50% recombination, the two genes are on the different chromosomes. The double crossover, when there are 2 points or 2 traits are taken into consideration, and the progeny of single and double cross are shown here in this diagram with parental and recombinant type with single crossover and double crossover example. The examples and the figures are repetitive of the fact that there are new genetic constitution taking place in the prophase 1 of the meiosis. The 3-point crossover is the final and the ultimate crosses to form the genetic map, which are shown here in the parental types, the consequences of double crossover, and the 3-point mapping showing the test cross using a resultant progeny, the P purple, round and juicy type of fruits having 3 traits and showing their recombination and crossing over frequency, and as a result we see of the genes responsible for P, J and R characteristic in parental types and the recombinant type which is present in the lower side of the chromatid. The rewritten form of test cross is shown in this diagram for the characters P, J, R. The genetic map of the PJR region of the chromosome computed from recombination data shows the distance in the units of the recombination and crossing over, that is MU, and the 20.8 and 10.0 units have been shown as a distance between P, J and R genes.